Okay, well, let's make a start. Welcome again and good afternoon. Welcome to the Coffee Science Seminar Series. My name is Michael Advardi. I'm Professor of Legume Genomics here at the University of Queensland, and it's my pleasure to introduce April Hastwell today. But before we do that, I'd like to acknowledge country. So the University of Queensland acknowledges the traditional owners and their custodianship of the lands upon which we meet. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable contributions to Australian and global society. So just a couple of housekeeping items before we continue. Today's seminar is scheduled from 12 to 1, and we'll try to wrap it up by 1. After the presentation, you'll have an opportunity to ask questions through the question and answer tab. So be sure to use the Q&A tab and not the chat to put your questions forth, and we'll present those to April after her talk. Okay, so it's a great pleasure to introduce Dr. April Hastwell this morning or this afternoon. April is a new member of QAFFE, the Queensland Alliance for Agriculture and Food Innovation. April is a plant molecular biologist specialising in plant microbe interactions, legume nodulation, nitrogen fixation, and plant peptides. They are a GRDC Fellow and have previously held an ARC DECRA and a UQ Amplify Fellowship. Dr. Hastwell's research has uncovered key signalling peptides and mechanisms regulating nodule development in both crop and model legumes, and is now focused on translating these discoveries into practical outcomes to improve symbiotic nitrogen fixation and increase crop yield for growers. So, April, with that, I'd like to invite you to present your talk on enhancing nodulation through targeted editing of regulatory peptides. Thank you.
Thank you, Michael, for that introduction. So as Michael mentioned, today I'll be talking a bit about peptides and what role they play in legumes. And this is work that largely comes a little bit from my PhD that I did with Brett Ferguson and Peter Greshoff here at UQ. And now I'm using that to apply to crops per se in a project also with Brett and with Michael as well. There we go. So I too acknowledge the traditional owners of the land and their custodianship of the lands on which we meet, and pay my respects to their ancestors and descendants. So I wanted to start off by really kind of introducing peptides because it's a bit of a unknown space for many people. And essentially, though, they're just small proteins, so usually less than 100 amino acids long, although many of the functional peptides are much smaller than this, 10 to 20 amino acids. They're typically less than 10 kilodaltons in size, and this is traditionally kind of lower than the resolution of many of the techniques that you can use to extract these peptides, but we do have a lot better methods at the moment, which is making it much easier to have a look at the mature structure of peptides. They function at very, very low concentrations, so 10 to the minus 15 is femtomolar concentrations, and to date, very few peptides have actually been isolated at endogenous levels. Which again makes them a little bit difficult to work with. And so we typically rely on the genes that we can pull out of genomes. And again, with annotation pipelines, they're not really well annotated, and that's because they don't have typical domain structures, and they're below that threshold of many pipelines pick up. And not only that, where those genome annotations are coupled with things like RNA-seq, they also sometimes are missed because they're very specific to a particular tissue or they're very specific to a particular stimuli. And so we sometimes don't see their expression very, very easily. And there's so many different types of peptides. There are cysteine-rich peptides, so they have disulfide bonds. There are short open reading frames that are present within the genome or within regulatory elements of other proteins. There are peptides which are derived from functional precursors. So these are excised from a larger precursor and then they perform a different function. There's also peptides, unmodified peptides, which don't have anything for what we can tell. And the thing that my research largely focuses on is these post-translationally modified peptides. So these usually have a larger precursor and then they're cleaved to a much smaller functional peptide, which is often post-translationally modified through things like hydroxylation and glycosylation, so sugars are attached to it, or in some cases there is a tyrosine which is sulfated. So this is what I've been focusing my research on. They're quite fascinating from an evolutionary perspective as well. What we tend to see is over time, you get more physiological adaptations, and with that, we see more diverse peptide families. And within each of those families, you also see an expansion as well. So for clear peptides, which are the earliest, one of the earlier peptides that kind of came about in early plant species such as algae, you tend to see only a couple of copies, whereas in more advanced plants like flowering plants, that gene family has really expanded from 2 to something like 40 to 80. There's quite a lot of tissues that they can function in. And of course, you know, with those special with different organs coming about as well, you get very specific peptides which are involved in floral abscission, for example, which is this Eda family of peptides. So they're very, I find them fascinating from an evolutionary perspective, and that has driven some of the research stuff that I've been doing. And just a kind of a snapshot of what they actually do in the plant. You pick your favorite organ or kind of biological process. There's really a peptide that does it. So that's involved in those processes. So things like floral development, floral abscission, I've already mentioned, things like fruit shape, seed and seed pod development, response to biotic stresses. So these are both pathogenic and beneficial microbe interactions, as well as being involved in insect responses. In the soil and of course in the atmosphere, they do respond to a lot of nutrient stresses or even the presence of the nutrient being there, and drought stress as well. So one of the neat things about peptides is that it really, that signals in the root can travel to the shoot, and it allows that communication between the plant organs. So they're key signaling molecules to kind of maintain homeostasis as well. They're also involved in root development, leaf development, and shoot development as well. So things like stomato opening and closing and whatnot. There's a lot in meristem maintenance, making sure that meristem, there's proliferation and differentiation as well. So I'm really interested in those kind of root specific peptides, but knowing that they are also those peptides that enable communication with the shoot as well. And so this brings me to look at symbiotic nitrogen fixation, because it's a really unique interaction between legumes and a bacteria that lives in the soil called Rhizobia. And what happens is after you get, you know, that unique communication between the plant and the bacteria, you get these novel organs being formed on the root called nodules. And inside those nodules are Rhizobia, and the Rhizobia have an enzyme called nitrogenase. And the nitrogenase converts atmospheric nitrogen into a form that the plant can use. And in exchange, the Rhizobia receive sugars from photosynthesis. So this is where that carbon is being exchanged with the shoot. And also this nice little niche area to live in. And what's kind of interesting is you've also got leg hemoglobin, which takes away the oxygen from that nodule, and it also gives it very distinct pink color. a little bit like animal hemoglobin. Of course, symbiotic nitrogen fixation is really, really important for the environment and for agriculture. It contributes $3.5 billion worth of nitrogen to Australian soils. And because of this process, we're able to alleviate some of the impacts, the environmental impacts of synthetic nitrogen fertilizer. So we don't have to apply quite as much. And we kind of capitalize on this by using legume crops as either in intercropping or in rotations to try and bring some of that nitrogen back into the soil. And it's quite a complex process because, you know, you've got a lot of carbon, nitrogen moving around, you've got nodules being formed, you've got immune responses to enable that bacteria to enter the plant. And so as a result, there are a lot of molecular pathways and genes that we know that regulate this symbiosis. And currently there's more than 200 genes that we know of in nitrogen fixation. And plant peptides play a really large role in a lot of these signaling pathways. So there's peptides that are involved in regulating the number of nodules, whether the plant actually is infected by the Rhizobia in the first place. You've got peptides being involved in actually forming nodules and maintaining that meristem within the nodule as well. And then there's peptides, involved in bacteroid differentiation. So that Rhizobia is free living in the soil. And when it goes into the plant, it has to change into a bacteroid, and that allows it to turn it to actually fix atmospheric nitrogen in that form. More recently, there are some peptides that we know involved in nodule positioning, so where the nodules are actually located in the root, as well as understanding the plant's own internal nitrogen status and maintaining that nitrogen ion balance. And that's kind of, perhaps there's a link with the leg hemoglobin there. And so my research largely focuses on those peptides that are involved in regulating nodule number. So I'm going to talk about two peptide families that I largely work on, and the first one of those are clepeptides. And we've known about clepeptides for many years, and the pathways that are involved in clepeptide signaling, I think it's more than 25 years. And so we have a good understanding. So I'm going to tell you a little bit about the background and kind of where I'm going next with this fundamental knowledge, which is quite exciting. So this is the pathway that regulates nodule numbers, and it's called autoregulation of nodulation. And so after you've got the plant communicating with the rhizobia, you've got some cell division starting that enable nodule formation, and then you've got the production of two clepeptides, RIC1 and RIC2. And these are rhizobia-induced clepeptides. They are part of those post-translationally modified peptide family or type, and they cleave to a functional 12 amino acid sequence. They have hydroxylation, and you can, on the seventh proline there, they've got 3 arabinose sugars attached to it, which is attached by an arabinosyl transferase. And this particular, the way that these sugars are actually there is really unique to plants. So it's a little bit difficult to synthesize, and there's some specialized glycochemists that we have to get involved with to collaborate with, to have these synthesized for us. So these clepeptides are produced, they are transported in the xylem, and they interact with a leucine-rich repeat receptor kinase. And this is called NARC, and NARC acts as a homodimer or a heterodimer. And then from there, you get a decrease in the level of a microRNA called microRNA 2111. And this microRNA is an inhibitor of an inhibitor. So when it's there, it enables nodulation to progress. And then after you get those nodules forming, you've got the clepeptides, and that downregulates the microRNA, so then nodulation is actually inhibited. And so it's quite convoluted, a little bit more complex than we initially thought, but this kind of feedback and this feedback loop enables the plant to inhibit or prevent too many nodules from actually being formed. So why does a plant need to actually regulate nodule number? It's important, I guess, agronomically because nodules are where symbiotic nitrogen fixation actually occurs. So it is one of those players in how successful it is, along with things like the bacterial strain, soil condition. There's a whole lot of other things that people are looking into as well, but nodule number is one of those things. And so when you've got no nodules, what you end up with is in a soil that's deficient in nitrogen is you don't have enough nitrogen to produce things like chlorophyll. So you get yellow plants there in plants that can't nodulate, the nod minus plants compared to the wild type, which you see in the middle. And then on the far side there, you've got a plant which actually can't control how many nodules it forms. And we call this phenomenon supernodulation. And if you were to pull up one of these, and this is bean and soybean, but if you were to pull up a supernodulating plant, what you see is really an excessive number of nodules being produced. And this really affects the shoot as well, because you get, in a wild type, you have, that carbon nitrogen balance is maintained, or in a supernodulating line, what you get is too much carbon being redirected from the shoot biomass and yield, and it's being redirected to maintain nodules and to maintain that bacteria that reside within those nodules. And mutations in that core supernodulation include that receptor of the peptides NARC, as well as things like too much LOVE and the enzyme, the rapinosyl transferase, which adds those sugars onto the peptides. So this is not an ideal situation. But it's this particular pathway is really highly conserved across all legumes. This work has been around for 25 years, including Peter Greshoff, who's been here identifying supernodulating mutants. And you can see there, so that's the NARC mutant in bean, has that supernodulating phenotype in lotus, as well as in metacargo there. And they all impact the root. But this mutation is we see that across many crops. And to investigate this a little bit further, one of the things we did to say, well, how conserved actually is it? And so in my PhD, we had a look at these. We used soybean RIC1 and overexpressed that in common bean in both the wild type and the supernodulating mutant. Oh my, it's a little bit stuck. Sorry. And the supernodulating mutant. And we were able to see a decrease in nodulation when you overexpress the soybean cleve in bean, indicating that it is highly conserved. Now, soybean and bean are very closely related. Their genomes are very, very close. So we took that a little bit further, and using some synthetic peptides and adding it to the shoot, which is where the peptides actually function, using PDL feeding in pea, we're able to also decrease the number of nodules using that synthetic peptide. And this again is in a NARC-dependent manner, which I haven't shown here, but it's a really, really highly conserved mechanism. So it enabled us to use what we know about one particular legume and adapt that to other crops quite easily. So there's been lots of fundamental research in this space by many groups around the world, and one of those groups is in China, our colleague Yu Feng, and they've actually taken this a little bit further and found a way to use this to our benefit by creating mutations not in the receptors and downstream signaling pathways, but those cleaves themselves. So they made edits in, there's two Rhizobia-induced cleave peptides, and they made combinations of these edits. And so in this graph on the corner there, you can see nodule number per plant. And in the wild type, you've got around 100 nodules compared, and then you've got that non-nodulating mutant. And then when you make a combination of edits, what you get is a stepwise increase in nodulation. But it's not to the same effect that you see with that receptor there, which is really quite exciting, because when you have a look at the shoot, you also see an increase in biomass as well. And they took this even further and they put this into the field in multiple sites in multiple years. And they actually saw an increase in biomass, increase in yield. And as a result, they saw an increase in symbiotic nitrogen fixation. And so instead of having too much carbon being redirected to the roots because of those added nodulation, that increase in biomass in the shoot was able to maintain that those nodules within the root. And so in essence, they optimize nodulation. So I wanted to know, well, can this be achieved for other legumes? It's quite an exciting find. And so when I was thinking about a research plan for GRDC Fellowship, I thought, well, chickpea would be a good target for that. Australia is the largest exporter of chickpea and the second largest producer globally. And more recently, there's been even more money available for chickpea because I know that India had some issues with diseases, and so they reduced the tariffs, and so we were able to export chickpea to India as well. So it seemed like a good fit. We're kind of tackling that. Michael Avadi is also kind of looking into optimizing nodulation in a different way than me. So I'm going to use genetic variation induced by a CRISPR, and so using tissue culture to create edits in these clear peptides and to see whether we can increase nodulation and as a result yield. And Michael, along with many colleagues around Australia, are looking at different legumes to see whether this type of mutation already exists within the population and whether we can actually harness that. And so, you know, it might be possible that we can achieve this optimized nodulation using a couple of different methods. So I wanted to step back a little bit from nodulation because clear peptides are, you know, one of the most well-characterized peptide families, and they know all about in everyone's least favorite plant, Arabidopsis. But we've learned a lot. They are involved in the root apical meristem in maintaining that stem cell population there, in vascular development, in the shoot apical meristem, which is where clavata 3 is. That's the namesake of the clear peptides, the first one identified. They're involved in ABA signaling to regulate stomatal opening and closing in response to drought. So that's that communication with the root there in floral development. And there's one clear peptide, or clavata 3 in tomato, which is quite an interesting case because they actually worked out that a mutation in the promoter of clavata 3 in tomato is what resulted in our larger tomatoes that we see and is really involved in the domestication of tomatoes. So it already has agronomic potential from there. There's also nodulation down there, which I've spoken about. One of the biotic processes, which is really quite linked to nodulation, and they share some of the same regulatory pathways, is mycorrhizal symbiosis. Of course, a lot more plants can form a mycorrhizal symbiosis, and so there might be a little bit of potential there to optimize clear peptides in that space, a little bit in pathogens as well. So you can see they function in quite a few pathways. In saying that, that's really probably only a fraction of what they do, because legumes contain more than 45 clear peptide encoding genes each. So it ranges, you know, soybean has 80 because it has a duplicated genome, but for the most part, it's between 40 or 50 peptides in each of these species that I've looked at. So soybean, chickpea, which is more recent, pea, metacargo, and lotus, and bean. And this is some of the work to identify these peptide families that I've been doing and building on previous research in this space. And it's enabled by a lot of the more advanced genome annotation that has come out. And knowing that the clear peptides are involved in a lot of different processes, some colleagues, as well as students in the lab, are looking at how they're involved in not only nodulation, but a lot of other abiotic and biotic interactions as well. And Alex also, and I'm aware the banana is definitely not a legume, but Alex has been working on identifying clear peptides in banana and having a look at their role in fusarium welts as well. So there's lots of opportunities to find ways to use peptides as a means to improve plant growth and yield and interactions with their environment. So I'm going to switch a little bit and focus on what I've actually been doing for the last 5 years, which is looking at another peptide family, which are root growth factors. So these still belong to that post-translationally modified peptide family, but where clear peptides tend to promote meristem differentiation, the root growth factors promote proliferation of the meristem. Yeah, that's what I've been looking at. And so the research for this particular family is much newer. It's not as well advanced, but there's some exciting opportunities to kind of look at plant development as well. So when I was thinking about this project for my DECRA and I wanted to look at peptides, and I wanted to look at peptides in nodulation, and there are a lot of genes, you know, that are very unique to nodulation and legumes that enable it to progress. So I mentioned, you know, there are over 200 genes that have been identified so far, and this is some work from Michael Advadi's group as well to put together which legumes have, you know, specific genes to enable nodulation. And knowing that, coupled with the fact that there are genes present in the legumes that if you don't have it, if it's knocked out, then it completely abolishes nodulation. So any kind of nodulation gene is usually very specific to legumes as well. And so then I had a look at the literature, and I came up with these root meristem growth factor peptides that were first identified in regulating the meristem of the root apical meristem, but also lateral root development. And I'll just walk you through the pathway that we know of so far. So you've got that prepropeptide, which is cleaved. It's hydroxylated at that proline and sulfated at a tyrosine. It is perceived by a root growth factor receptor, which is also a leucine-rich repeat receptor kinase, very similar to NARC, actually. And then you've got a whole cascade of downstream signaling events that really interplays with auxin, and it has effects in, you know, gravitropism, those types of developmental aspects. And then within the last 5 years or so, we now know that they play a larger role, not just in the root, but also leaf immunity and floral development as well. And so this was my target family to see whether they had any role in nodulation. And so I had a look at the genome. There's a lot of manual curation involved, but I went through it and I identified in soybean 28 root growth factor genes. So this equates to about 10 to 14 in other species, in other legume species, I should say. And there's a lot of systematic phylogenetics to identify clades, which are very specific to legumes. So you include legumes and non-legumes in phylogenetic trees, a little bit like this. And coupled that with having a look at existing RNA-seq data and performing real-time PCR to have a look at gene expression in response to inoculation. And this was to identify genes that could possibly be involved in nodulation. And we came up with 5 candidates that seemed to fit this kind of criteria of having may be involved in nodulation. And so of those 5 genes, we had a look at where they're expressed using promoter gut study. And this was some work done by my postdoc at the time, Shi Tong, and my PhD student, Yuhan, who recently graduated. And they had a look at where these genes were expressed. And you can see that They're involved in definitely nodule development, as you can see by the blue color there, and particularly in mature nodules. And this was really reflected when we had a look at expression, real-time expression data as well. You can see that it's expressed at 14 days after inoculation, so when the nodules are starting to mature. And they're really, really highly expressed in nodules compared to just root, uninoculated root tissue as well. And so I've only got 2 genes up there, but all 5 genes had very similar expression patterns in terms of being expressed in the nodules, but also in the root as well. So this is what those 5 genes of what I've been focusing on. So we were able to synthesize the mature root growth factor peptide that had that sulfation, and when you apply that to soybean roots, you saw a decrease in nodulation. So this essentially negatively regulating nodule development. And there is a change not only in nodule number, but also the number of mature nodules and immature nodules that are on the plant as well at different concentrations. And then the next step was to overexpress this using Agrobacterium rhizogenese mediated hair root transformation, which enables, it's kind of like a chimeric transformation system that is a little bit quicker than stable transformation. And we noticed an interesting phenomenon that, at 7 days after inoculation, when you overexpress the peptides, you completely almost abolish nodulation. But over time, some of these plants which had overexpressing particular peptides were actually able to recover. So perhaps it's delaying the nodule formation, or it has a role in nodule maturity, because there's also an impact on how many of these nodules actually mature to fully functional nodules that contain rhizobia that fix nitrogen. Of course, having a role in root growth generally, we did have a look at the root, and we get these really interesting phenotypes because it messes up the auxin distribution within the plant, and you get these curly, wavy roots. As you can see here, when you overexpress the peptides, and when you apply synthetic peptides, you get a decrease in the root biomass. And this is really reflected on the fact that the roots have less finer roots, which is what you can see here. So there's a decrease in the number of fine lateral roots that the plant is actually producing. So we do have a change in root development and nodule number, and we've had a kind of look at the interaction of those as well. So then the next step is to create mutants, which is quite difficult to do in soybeans. So we opted to do, again, CRISPR in hair root transformation, which is not an ideal system, but we got some preliminary data, which is really quite exciting to say that, the opposite of overexpression and peptide analysis and peptide application, you get an increase in nodule number per root, which is what you can see here. But more surprising is we get this kind of increase in root length and increase in kind of overall root growth as well. And that's kind of something that could have some agronomic potential, which is quite exciting. And so at the same time that I was doing this, someone who worked with Michael Avati actually was working on these peptides in metacargo. And so we also know that they play a role in many species in nodule development. All right, so I'm kind of coming to the end, but I wanted to leave you with this. So I've spoken about clear peptides and root growth peptides, and one of them, we have a lot of fundamental information, and we're now into that stage where we can actually modify or modulate how much of these peptides there actually are for impacts in yield and root development and nodulation to benefit crop production. And the other one that I'm working on is, it's much more kind of in its infancy in really, but I think there's some potential as well there to improve crops using those peptides. And so I'm also looking at that in my GRDC project as well. And of course, if anyone ever wants to chat about peptides in their favorite developmental process, I'd be happy to talk. Sometimes they're not always easy to pick out of the genome, and they're often missed. And they're not well annotated, even if they are predicted within a genome. So be happy to chat there. I've spoken very fast. I apologize, but I'm already done. So thank you. There's a lot of people who have contributed, Michael and Brett, to the current GRDC project, and Millie as well for her advice, and then some collaborators, Dougal, Struthy, and then everyone who's kind of forms the larger integrated legume research group. Thank you.
Thank you very much, April. A great tour of CLEs and ODFs and their role in plant development. So just a reminder to folks to use the Q&A tab for questions. I'm looking for some here, but I'll start off, I think. April, I had a question about whether you think there might be a trade-off between nodule number and nodule size. So some of what you're aiming to do is perhaps moderately increase nodule number. What might that do to average nodule size and volume, and perhaps nodule activity? Do you have any information about that? Any potential trade-off?
Good question. It's a good question. So in supernodulating mutants where there's too many nodules, the nodule size tends to be much smaller. And as a result, I think per nodule nitrogen fixation is obviously reduced, but overall it's increased. So I don't know that if you increase, well, I think they have shown that if you increase that nodule number just a little bit, then you do get just enough to carbon to support an increased nodule size. I don't know about the size per se in the RIT mutants. It'd be interesting to look at, and I think, yeah, no, it's a really good point. I don't know what the trade-off is for the CLE mutants in terms of size and nodule number, but yeah.
I think it's one aspect of the GRDC project that we're running, that you mentioned, that I'm leading. We will be looking at those sorts of things, you know, the influence of size versus number, and of course other things like biochemistry and nodule metabolic processes. So in a sense, that GRDC project that you mentioned is a lot more diverse and sort of open-ended than your very focused project. Please. And anyway,
but I think you know, once I've got the transformation kind of a proof of concept, and then if there's any genetic variation that you found, then perhaps we can come back to transformation and help there. Yeah,
they're very complementary.
Yeah.
I have a couple of questions now. Eloise Fu in Tasmania.
Hi, Eloise.
Do you think peptide application is a viable future agrochemical?
It's complicated. I think that it definitely could be. Currently they're not stable enough to be an agrochemical, and they're very expensive to make. I think if you were able to increase the stability, so I know that there's some cyclic peptides, for example, and perhaps that could be a method of delivery to add, you know, these kind of functional peptides on it. I do think there are too many potential off targets of a given peptide, because the functional domain of a lot of them within a same family is very, very highly similar. And so I think there could be some issues there. Not to say it's out of the question, but I think it's definitely we need better technology to create it and look to do that.
Thanks, April. I don't know if everyone caught what you said about cyclic peptides, but it's a way of maybe stabilizing peptides that are in their linear form are susceptible to proteolysis. So
Yeah, so
cyclization
cyclopeptide.
maybe stabilizes them. And
Yeah, yeah, definitely. So
right,
that's
very good. Karen Prieto asked, does peptide application show some above ground effects? So are you getting above ground effects as well?
so in our experiments we didn't, but the level of peptide maybe perhaps if you had applied more, we could have seen an above ground effect. It did definitely have a change in root development, and it was, if there was, it was very, very subtle. I think that the amount of peptide that actually gets into the root and is actually perceived is probably very minute compared to what we applied. So perhaps if you applied a larger concentration, you might have seen an above ground effect as well. But it's definitely something that we do keep an eye on just to check.
Well, you mentioned that some of the peptides affect shoot meristem activity. So are there experiments in other species that look at aerial application and impacts on shoot architecture, for instance?
I am not familiar with any, because the clear peptides actually, when you apply them, they tend to stop the proliferation of cells, and so you get arrested development example. So you don't really want that in your crop, and so that's why that mutation in the clavata3 makes bigger fruits there, because it enables that proliferation of cells, which but yeah.
Great. Damien Hine has a question.
Yeah.
He asks, where do you see the biggest industry translation and possible commercialization opportunities that might emerge from your work?
Yeah, I think the generation of lines that have edits in peptides is definitely something that can be commercialized. There are so many, and what's really nice about peptides is that they tend to be, well, some of them tend to be a little bit more specific in their function. So if you knock out a receptor, for example, you get a much wider range of phenotypes. But because the receptors, because the peptides are a little bit more specific and you've got a little bit of redundancy in those pathways, if you edit something, it just might help the plant just a little bit. And so I think gene editing is definitely up there with commercialization, and of course adding peptides isn't out of the scope, but I think improving nodulation, improving other abiotic interactions and biotic interactions. There's definitely, there's so many peptides that the opportunities are really quite endless to see what they can do.
An attractive feature of them is that they operate at the cell surface, I guess, or many do, if they're effectors that interact with signaling molecules. So given that, and despite the fact that they might not be stable, if you were to take an approach like cyclized peptides, do you think creating a library of random peptides might be a useful approach to, look at effects on plants that might be stable and deliverable and also cheap, because these things operate at such low concentrations?
It's definitely possible. Yeah, I think it's definitely possible. I mean, other than the root growth factors aren't too bad to make. The commercial companies can make those, but making the triarabinose is a little bit difficult, the building block. And we also, I don't think people who work in this space, we know that the enzyme that attaches the first arabinose to the proline, but I think there's another enzyme involved. And so perhaps if we're able to identify those enzymes, then maybe we could do it in a more synthetic system and actually, make these on a larger scale rather than relying on chemical synthesis. But yeah.
Do we know what sugar moieties or arabinose addition does to the or what's the function of those additions? Is it required for receptor recognition, or is it more to do with systemic transport of these things or export from the cell? What do we know about these modifications in
That's
terms
a good
of their
question. The number of ribose sugars does change the binding efficiency to its receptor. And so, you know, a peptide, a click peptide with 1 arabinose has a lower binding efficiency than one with 3. And we did do a little bit of work in that space, just having a look at what 1 arabinose sugar does, and we can see that as well, with kind of phenotypic effects as well. The effect, it doesn't have any effect on transportation. I know that there's a glycine right in the middle, and so perhaps it changes the conformation of the peptide and allows, you know, maybe a little bit more bending to enable receptor recognition. It doesn't have a role in transportation. What we do, however, find though is in these post-translationally modified peptides, the ones that we know are able to move systemically tend to have a couple of extra amino acids at the end of the peptide themselves, that what we don't call part of the functional peptide, but perhaps they're the ones that are first cleaved off, you know, as it's traveling throughout the plant, and so you're left with that functional peptide at the end.
Right. Well, I think we'll take one more question, and again, it's from Karen Prieto. Do you think these novel root peptides act through the known AON pathway? So these novel peptides, the root peptides, the RGFs, I guess.
Yeah, so we did some work and overexpressed the root growth factors in the NARC mutant to see whether there was any kind of, you know, if it was still able to suppress nodulation in the same way, and we did see that. So I think it's either, you know, it maybe there is some interaction, but it's not direct. If you have a look at the NARC receptor family, NARC and similar things like the root growth factor receptor, they're very similar, but they're not quite the same. So I don't think one can perceive, like NARC can perceive the root growth factors. But I from our work haven't seen any direct link with them. But it's trying to figure out where they do fit in the overall pathway, I think is something that needs to be done. And perhaps it's definitely something that needs to be done. There's a lot more questions than answers for the root growth factors.
Right. Well, we'll leave with your own questions.
Yes.
There are no more questions from the audience, so thanks to those that have listened and participated with questions. Appreciate the discussion. Thanks again, April, for a lovely seminar. I think we've got a couple more slides. The next seminar in this series is on July 15. We have a guest from Texas Tech University, Professor Jagadish Krishna, who's going to be talking about the use of idiotype breeding in sorghum to deal with some environmental stresses, I guess, such as drought. So stay tuned and join us again. If you want to learn more about our science seminar series program at Coffee, I think you can get that using this link here. So and I'm sure you can find it in other ways through the Coffee website. So stay tuned for future seminars. And again, April, thanks again for a lovely introduction to your work, and welcome again to Coffee, and all the best in your research.
Thank you, Michael.